Bismillahirrahmanirrahim. Dear students, Assalamu alaikum. In our previous lecture, we have discussed about the cell cycle, in which we have discussed that cell cycle completes in two main phases. First one is the interphase and second one is the M phase, which is also called as mitotic phase. In previous lecture, we have discussed about the interphase. Today our topic is M phase. M phase is further divided into two main subtypes. First one is mitosis and second one is meiosis. In our today's lecture, we are going to study about mitosis. Mitosis is a type of cell division in which one cell divides to produce two daughter cells in such a way that number of chromosomes remains constant in the daughter cells. For example, look at this. In human cell there are 46 chromosomes. When this cell divides to produce two daughter cells, the number of chromosomes in these daughter cells remain same, that's 46. So it is the mitosis in which the number of chromosomes remain constant. Mitosis is again a lengthy process, so it is further divided into two main phases. First one is called as karyokinesis and the second one is cytokinesis. Karyo mean nucleus, kinesis mean division. So karyokinesis is the division of nucleus. Second step is the cytokinesis. Cytokinesis, cyto mean cytoplasm and kinesis mean division. So cytokinesis is the division of cytoplasm. In first step, we will discuss about the karyokinesis. Karyokinesis is the first and lengthy phase of mitosis. So it is further divided into four main subphases. The first one is prophase, second one is metaphase, third one is anaphase, and fourth one is telophase. Prophase is the first phase, and in this phase the main changes occur are condensation of chromatin into chromosomes, division and movement of centrioles towards opposite poles, formation of spindle and ester fibers, which is also called as mitotic apparatus. In case of plants, mitotic apparatus is not consisting on ester fibers. The spindle fiber directly originate from cytoplasm, while in case of animal cell, spindle fibers always originate from centrioles. Next one is the disappearing of the nuclear membrane and scattering of chromosome on the spindle fibers. The next phase is metaphase. In metaphase, the main changes are arrangement of chromosomes on the spindle fiber at the center or equator of the cell. Next one is the arrangement. Chromosomes are always arranged individually on the spindle fibers through the specific regions which are called as kinetochores. So this is the main change which takes place in metaphase, that is the arrangement of chromosome in the center of the cell and forming mitotic plates. The next phase is the anaphase. During anaphase, the main change is the contraction of spindle fiber, which exert pulling force on the chromosomes. Due to this reason, chromosome divides from the centromere and starts moving toward the opposite poles. The next and last phase is the telophase. Telophase is the reversal of prophase. It means the things, the structures which are produced during the prophase, now they will finish or they will be disappearing, and the structure which disappear in the telophase, they will reappear in this phase. For example, first thing is the reaching of chromosome at poles. After reaching the chromosome, the next one is the disappearing of spindle and ester fibers, which, as you have already studied, are produced during prophase. So these disappear in this phase. Next one is the formation of nuclear membrane, which disappeared in the prophase. So due to the formation of nuclear membrane, one nucleus is divided into two, and at this stage the process of karyokinesis completes, because karyokinesis is the division of nucleus in which one nucleus divide into two. The next phase is the look at the diagrams. For example, this diagram is about the prophase. You can see that chromosomes are formed due to the condensation from chromatins. These centrioles are starts moving toward the opposite poles, formation of spindle fiber, ester fibers. All these changes takes place during the prophase. Next one is the metaphase. In metaphase, chromosomes are arranged in the center at equatorial plate, as you can see. The main change during metaphase is only the arrangement of chromosome on the spindle fiber, and individual chromosomes are arranged. The next one is the anaphase. During anaphase, the chromosome starts moving toward their poles, and this movement is due to the pulling force of the spindle fibers. The last stage is the telophase. In telophase, chromosomes reach their poles, spindle fibers and ester fibers disappears, and nuclear membrane is completely formed. In this way, one cell is completely divided into two daughter sorry one nucleus is completely divided into two nuclei. The next stage is the cytokinesis. Cytokinesis is the division of cytoplasm. In animal cell, cytokinesis takes place by the invagination of cell membrane from the equatorial region at which chromosome was arranged in the metaphase stage. At that region, deepening of furrow and meeting in the center of the cell membrane takes place, which completely divides one cell into two complete cells. Again, in animal cells, the cell membrane invaginate from the center, the invagination deep, and ultimately the cell membrane meet in the center, and one cell is divided into two daughter cells. While in case of plant cell, there is the formation of a special structure which is called as phragmoplast, because the outer covering of plant cell is cell wall. Cell wall is rigid and hard structure. It does not invaginate. So due to this reason, there is the formation of phragmoplast. Phragmoplast is basically formed by the deposition of special structures which are called as Golgi vesicles. These Golgi vesicles fuse together and they form the central cell plate, and after that cell plate, one cell is completely divided into two cells. For example, you can see in the diagram that in case of animal cell, there is the formation of invagination or furrow from the center, and it completely divides one cell into two cells. While in case of plant cell, there is the formation of Golgi vesicles. These vesicles combine together to form the cell plate, and ultimately one cell is divided into two daughter cells. So this is all about the mitosis. Hopefully you understand it. If any query, you can ask me at my phone number. uh, in next lecture, inshallah, we will discuss about the importance of mitosis as well as about the few stages of meiosis. So in our next topic, inshallah, we will discuss about the meiosis. Thanks. Allah Hafiz.